Welcome to zoology class. In the last few classes, we have been discussing about single gene mutations and the disease caused by these diseases. These are called Mendelian disorders. So we are discussing about some of the Mendelian disorders caused due to single gene mutation. Okay, while discussing about mutations briefly, we have seen that mutations are of two types. There are gene mutations and chromosomal mutations. So far we have discussed some of the disorders caused due to gene mutations. Of course, there are chromosomal mutations also. So chromosomal mutations are of two types. Chromosomal mutations due to a structural change in the chromosome and chromosomal mutations due to a change in the number of chromosomes. Structural change in the chromosome may be due to deletion, insertion, or substitution of chromosomal parts. So that may affect several genes present on that segment of the chromosome concerned, and that may lead to a very serious disorder, or sometimes maybe the viability or the existence of the individual may be questioned. And another one that we are going to discuss in this class here is mutations due to change in the chromosome number. And mutations due to change in the chromosome number again is of two types. One is polyploidy and the other one is aneuploidy. Let us see what is polyploidy. You are familiar with that? Generally, the organisms are diploid. We are all diploid organisms. So what do you mean diploid organism? Diploid condition. Every set of the chromosome has two chromosomes. For example, we have 23 sets of chromosomes. Each set of the chromosome has two chromosomes. The first set has two chromosomes, second set has two chromosomes, third set has two chromosomes. Like all the 23 sets of chromosomes are constituted of two homologous chromosomes. So that condition is called a diploid condition. In some cases, due to mutation, due to some changes, due to some reasons, this chromosome number may be changed. That means the whole set of chromosome may be increased by one chromosome. That means the first set, instead of having two chromosomes, may have three chromosomes. The second set also have three chromosomes. The third also three chromosomes, fourth three chromosomes, fifth three chromosomes. So whatever may be the number of sets of chromosomes, all the sets of the chromosomes may gain one more chromosomes, and the condition then is called triploid condition. So instead of diploid condition, every set when acquires one more chromosome, that condition is called a triploid condition. Then again, sometimes instead of acquiring one chromosome for every set of chromosomes, two chromosomes can be acquired. So the first set can be with 4 chromosomes, second set also with 4 chromosomes, third set also with 4 chromosomes, like that. Whatever may be the number of sets, all the sets of chromosomes will have 4 chromosomes each, and that condition is called tetraploid condition. Tetraploid condition. So these triploid condition and tetraploid condition are examples for or instances of polyploidy. Polyploid. So polyploidy is not very common among animals. But in plants, in certain plants, polyploidy occurs, and as a result, one species of plants suddenly, all of a sudden, may be changed to another species. Such occurrences are there among plants. So that is about polyploidy. We are not discussing in very detail about polyploidy here. And the second instances of chromosomal mutation due to change in the number or numerical change is aneuploidy. What is aneuploidy? Aneuploidy is also a change in the number of chromosomes, but here the number of chromosome is changed in a particular set of the chromosome, not the whole set. The number of chromosomes is not increased or decreased for the whole set of chromosomes, but a number of chromosomes increased or decreased for a particular set of a chromosome. This is called aneuploidy. Aneuploidy. And when we say about increase or decrease the number of chromosomes for a particular set of the chromosome, a chromosome number can be increased or decreased of a particular set. That can be, the chromosome number can be increased by one chromosome or it can be decreased by one chromosome. These are the usual occurrences. Suppose the chromosome number is increased by 1 for a particular set only, that condition is called a trisomy condition. It's called a trisomy. It's called a trisomy. Tri means 3. And if the chromosome number is reduced by one chromosome, that is called a monosomy. Let me explain it further. The normal chromosome condition, the normal chromosome number is diploid. Okay. Suppose for a particular set, say for example, the 21st set of chromosomes. So we have 23 sets of chromosomes. Suppose the 21st set of chromosome, instead of having 2 chromosomes, sometimes we have 3 chromosomes. Then we say that's a trisomy, and as it is occurring to the 21st chromosome, then we say it's the 21st trisomy. 21st trisomy. Suppose a particular chromosome loses one chromosome. Instead of the diploid chromosome, diploid number of chromosomes, it may have only one chromosome. Instead of 2 chromosomes, it may have only one chromosome. One chromosome is lost due to some reason that we will discuss. And such a case is called monosomy. So trisomy and monosomy are the two numerical changes happening to a particular set of the chromosomes. Now the question is, how should it happen? When does it happen? Why should a set of chromosomes gain one chromosome to become its trisomic? Or why should a set of chromosome loses one chromosome to become it monosomic? Trisomy or monosomy can happen both in autosomes and allosomes. So autosomal trisomy and monosomy are there. Allosomal trisomy and monosomy are also there. So this happens due to an error in the cell division. An error in cell division. Error in meiosis. Let us see how it happens. In a diploid condition, chromosomes exist in pairs as homologous pairs. During metaphase of the cell division, these homologous pairs align at the equatorial plane. And during anaphase, normally, these homologous pairs are dragged by contractile filaments, spindle fibers to the opposite poles of the cell. So out of the pair of homologous chromosomes, one homologous chromosome would move to the one pole and the other homologous chromosome would move to the opposite pole to become part of the daughter cells. This is what happens normally. But sometimes, due to an error in the cell division, these homologous chromosomes fail to be separated. They fail to be separated, and these homologous chromosomes move together to one pole. And the other cell and the opposite pole do not have no chromosome of that set. And the daughter cells formed as a result of this cell division would have no chromosomes for that particular set. Whereas the other daughter cell would get 2 chromosomes instead of 1. And this phenomenon, this condition is called non-disjunction. So failure of separation of homologous chromosomes during anaphase of meiosis is called non-disjunction. Failure of segregation or separation of homologous chromosomes to opposite poles of the cells to enter into the daughter cells is called non-disjunction. So what is the result of this non-disjunction? Let us take the case of a female individual with 2 X chromosomes. These 2 X chromosomes are the homologous chromosomes. So these X chromosomes along with other homologous chromosomes align at the equatorial line during metaphase. And normally during anaphase, one X chromosome should move to the one pole and the other X chromosome should move to the opposite pole to be part of the daughter cells. But due to non-disjunction of these X chromosomes sometimes move together to one pole and the other pole would not get any X chromosome. That means when gamete is formed, as a result of meiosis, gametes are formed. When gametes are formed, one ovum would get, or certain ova would get 2 X chromosomes and certain ova would not get any X chromosome. So what would happen during fertilization? The ova, suppose the ovum with the 2 X chromosomes fertilized with a sperm with Y chromosome, then the number of X chromosomes, the number of sex chromosomes would be 3 instead of 2. In the zygote, the sex chromosomes would be 3. Which are the sex chromosomes then? 2 X chromosomes and 1 Y chromosome. That condition is XXY condition. So that will result in an abnormality or a syndrome that we will discuss shortly after. Then what about the other ovum, which did not get any X chromosome due to non-disjunction? Suppose that fertilizes with a sperm with the X chromosome, then the zygote would get only 1 X chromosome. The homologous chromosome would be absent. So that also would result into an abnormality or a disorder. That also we will discuss in this class. Okay, so this is the same condition can also happen in the case of autosomes also. So trisomy and monosomy are the two abnormalities or two numerical changes coming under aneuploidy. Aneuploidy is the change in the number or decrease or increase in the number of chromosomes of a particular set. These are of two types, trisomy and monosomy. Okay. Now let us go to certain examples. Let us discuss certain examples of trisomy and monosomy. So as I told you already, both autosomal trisomy and monosomy are there. Allosomal trisomy and monosomy are also there. We shall first discuss about a disorder caused by autosomal trisomy. So the best example for this, or the most discussed example for this condition, is 21st trisomy. 21st trisomy. 21st set is an autosome. So it's an autosomal trisomy. So remember, we have 23 sets of chromosomes. First to 22nd are autosomes and the 23rd set is allosome. Okay, that may be XX in female or XY in male. 21st trisomy. That means the 21st autosome, the 21st set of chromosome has 3 chromosomes instead of 2 due to non-disjunction of the 21st homologous pair during anaphase of meiosis 1. Okay. How does it happen? 21st pair getting aligned at the equatorial plate during metaphase of anaphase. Instead of these homologous chromosomes going to the opposite poles and getting separated, they remain together. 21st chromosome, 21st homologous pair remain together and they go to only one cell. Okay. So when that forms a gamete, whether it is male or female, and when that gamete, let it be male gamete, fuses with a female gamete, a normal female gamete, with a 21st chromosome, and in the zygote after fertilization, the zygote gets 3 21st chromosome, 2 from one gamete and 1 from the other gamete. So such an individual will develop a disease, develop a disorder or a syndrome, and that syndrome is called Down's syndrome. What's a syndrome? A syndrome is a set of characteristic features of a disorder. It is called Down syndrome because it was discovered by a scientist called Langdon Down in 1866. That's why it is called Down syndrome. Okay. Now let us see what are the characteristic features of Down syndrome. These individuals are short statured. So their body stature, they are generally short people with a round head. They have a round head, their body is short, and usually their mouth is partially open. They keep their mouth partially open and with a protruded tongue. The tongue would be slightly protruded out. And this tongue generally would have furrows on it. Furrows or channels on it. And then they are mentally retarded. They are sterile people with a large palm, with a palm crease. A big crease, folding, small, very small, minute foldings or furrows on the palm. So these are the general characteristic features of Down syndrome. Let us repeat, let us say what are they? Once again, they are short statured, round head, partially open mouth with a protruded tongue, tongue with furrows, mentally retarded, sterile people, probably with a palm, large palm with a palm crease. As they almost look like mongoloid people, the condition is also called Mongolism. Okay. So that is about Down syndrome. Now let us go to a sex chromosomal trisomy. Sex chromosomal trisomy. I told you, trisomy can happen to the sex chromosome also due to non-disjunction of the sex chromosome. So here the condition is called Klinefelter syndrome. Klinefelter syndrome, because it was discovered by a scientist called Harry Klinefelter in the year 1942. Harry Klinefelter in the year 1942. That's why the disorder was named after him, Klinefelter's syndrome. Here, the chromosome pattern, as the non-disjunction happens to the sex chromosome, autosomes are normal. Normal number of autosomes are there. What's the normal number of autosomes? Normal number of autosomes are 44. We have 46 chromosomes, 23 pairs or 46 chromosomes are there. So 44 are autosomes or 22 pairs are autosomes and one pair, there is 2 chromosomes are allosomes. So 44 plus 2, 44 autosomes plus 2 are the normal chromosome number. Here in Klinefelter syndrome, there are 44 chromosomes, autosomes are 44, but sex chromosomes are 3 instead of 2. So we can write 44 plus 3. That 3 is XXY. So the chromosome pattern is 44 plus XXY. So they have 3 sex chromosomes. XXY. Okay. So now let us see the syndrome, characteristic features of this disease. They are males. People with the Klinefelter syndrome are males. But they are sterile. They are not fertile, they are sterile. And another feature is that they have a condition, they have a feature called gynecomastia. What is called gynecomastia? Gynecomastia means appearance of female characters, appearance of certain feminine characters or female characters in male. That is called gynecomastia. So feminine characters or female characters like enlarged breast, a lack of facial hairs. These are the two major features coming under gynecomastia. And generally these people cannot be distinguished from normal people. Apparently they have no problem. When they are dressed properly, they cannot be identified as people with Klinefelter syndrome. But they have gynecomastia and they are sterile. They are not mentally retarded. They have normal mental composure and physical stature. No problem with that. But the only thing is that they are sterile and they have gynecomastia. Okay. So that is about Klinefelter syndrome. And now let us go to the example for a monosome. So what we are discussing about here is sex chromosomal monosomy. Now this is called a Turner's syndrome. This condition is called Turner's syndrome because that was identified by a scientist called Henry Turner in 1938. The disease was discovered by a scientist Henry Turner in 1938. That is why it is called Turner's syndrome. Here, the monosomy is a sex chromosomal monosomy. Sex chromosomal monosomy. So, sex chromosome number is affected. Autosome number is same, normal, 44. So, here, one of the X chromosome is missing. The individuals are female. One of the X chromosome is missing due to non-disjunction of the sex chromosome. As we have already seen, female has two X chromosomes. Due to non-disjunction during the anaphase of meiosis, both the X chromosomes move to one pole and the other cell does not get any X chromosome. Suppose that cell without any X chromosome fuses with a sperm with X chromosome, then the zygote would get the condition X0. Only one X chromosome would be there. That came from the sperm. Okay. So such a condition is called Turner syndrome. They are generally females, but they are sterile. They are sterile females with a short stature. They are short individuals, sterile individuals with underdeveloped female sex organs. Ovaries and other female organs are underdeveloped. So they are sterile also. Short-statured individuals. The chromosome pattern is 44 plus X0. 44 plus X0. Okay. So let's see once again which are the examples once again for trisomy. We have two examples. One is Down syndrome. The chromosome pattern here is 45 plus XX or XY, because as the Down syndrome affects the autosomes, the sex of the individual has no it is not has any concern here. The 21st chromosome is 3 in number, so the autosomes totally increase by 1. Instead of 44, it is 45. So for Down syndrome, the germinal chromosome pattern is 45 plus XX or XY as the case may be. For next trisomy, sex chromosomal trisomy, Klinefelter syndrome, the chromosome pattern is 44 plus XXY. And for Turner syndrome, the chromosome pattern is 44 plus X. So, okay. So, these are some of the numerical changes under aneuploidy of chromosomes. Okay, we shall wind up the class here.